Thank you for attending our webinar. So today's topic is degradome sequencing for plant microRNA target identification. So for this topic today, we are going to do first is microRNA in plant, give a simple introduction about the microRNAs. And then we try to explain a little about the target identification. Then, most important, we want to introduce the degradome sequencing to everybody. And we will show some case studies. The last one we're trying to answer a question. So what do we know about the microRNAs? So we know first that the microRNAs are small non-coding RNAs. Usually, for mammalian, they normally is 20 to 22 nucleotide long, and for plant, normally a little bit longer. It's about 20 to 24 nucleotides. And all microRNA precursors normally form a well-predicted stem loop helping structure, and this do help us to do the prediction about the novel microRNA. And the photobic helping structure has very no free energies. So also this also help us to do the prediction. And we know that microRNAs are evolutionarily conserved. So from worm to human, from plant to mammalian, we do have microRNAs. And the regulation of microRNA is normally, we know, is binding to the complementary binding to the messenger RNA molecules. And they play as negative regulators of translation. And microRNA normally present in high copy numbers, and they are tissue specific. So if we put microRNA to the central dogma of the biology, we know that they regulate the RNA translation to protein. So the well-known microRNA database is miRNA-based, and we know that after Illumina release first the small RNA sample preparation kit, that means that we can use a high throughput sequencing method for the novel microRNA identification or discovery. We see a spike from microRNA on the microRNA discovery side. So right now we are using miRNA-based version 19, and it connected about 20,000 microRNAs entries, including 193 plant and animal and various species. And what's microRNA's function in plants? So as normally we know that microRNA regulate plant development, like what normally microRNA does for mammalian cell, and this development including leaf development and flower development and other developments. And the microRNA also involved in signal transduction, and for plant they also involved in disease and resistance, including biotic resistant and unbiotic resistant, unbiological resistance. microRNA normally involved in developmental stress response. And also microRNA regulate themselves, microRNA regulations, and also small interference RNA biogenesis and functions. So it seems that the microRNA play a very important role in plant in almost, if not the every point of the biological processes. So it's clearly that why we need to study microRNA in plants. So first we want to do the basic research and do the discovery, and also we want to try to understand, especially for plant, what's the stress response once the plant meet all different situations. And also it will help us to do the plant breeding, and we try to identify which plant or maybe which one for our need is the good one. Yeah. And the mechanism of the microRNA in plant. So for this topic, we know that the microRNA in plant is normally is near perfect complementarity to targets. So mammalia, we know that they just share 7 to 8 bases as complementary to the messenger RNA target, but for plant they are doing like nearly perfect. So that means almost all the bases are used for the complementary binding to the target messenger RNA. And upon binding to their target, messenger RNA target, the microRNA containing risks, I mean this including proteins, some proteins, they function as endonucleus. They will cleave the messenger RNA, and another function is that they will block the translation of the messenger RNA to the protein. So for the plant, so the cleavage site is normally precisely guided between the 10 to 11th nucleotide in the complementary region. This 10 to 11, 10 and 11th nucleotide is based on the sequence of the microRNA. So based on this, we know exactly where the cleavage happen on the messenger RNA, and also we can trace back the information to the microRNAs. So so first, we know that the microRNA gene will express a helping structure RNAs, and then this helping structure will be further processed by the DCL1 ribonucleus, and they probably will have also assisted by the HIN1 and the HYL1 proteins, and they will cut about 21 nucleotide long, and with 2 nucleotide 3' end overhangs. And then with an unknown helicase, another enzymes, so they will select one single strand of this double strand microRNA precursor, and this will incorporate to the risks with other proteins. And in the messenger RNA, at this moment, the messenger will be searched and bound to the risks, and the risks will search the messenger RNA, and then they will cut the messenger RNA, or maybe reduce the translation of the also uncleaved the target messenger RNA. Translation will be reduced. And here we need to pay attention to one thing, that is that after cleavage, the risks complex will leave, after cleavage will leave 5-phosphate on the 3' end fragments, so as shown here. So this phosphate will link to the 3' end. That means at this moment, this fragment will have poly-A tau at the 3-UTR region, after 3-UTR region, and also have a 5-phosphate at the 5' end. So this is very important for our protocol, for our degradation sequencing protocol. So we know that the microRNA function in plant is not just one-to-one. They play their role in the network of the regulations. I mean, messenger RNA or maybe protein, even pathway regulations. So that's important to let us know that if we just simply do the no throughput identification to find the target, we just see one spot of the regulations. If we do the whole microRNA identification, I mean for the whole microRNAs, and also try to identify whole network, we definitely need to go with the high throughput method. That's what we are currently using now. It's the small RNA sequencing plus degradome sequencing. That's the next generation sequencing method. So we are going to talk about the microRNA sequencing for a while. So we know that for the all sequencing projects, we can divide them, all these projects, to three major parts. One is the library preparation. That means we try to get the library for sequencing, and then definitely we go to the depth sequencing. That's the next generation sequencing method. And the major work will be on the data analysis part. Depending on different project, we need to adopt a different method for the data analysis. For the microRNA sequencing, normally we get like 5 to 10 million reads first. This is for microRNA sequencing. And after filtered all this kind of junk reads, we call it like adapter only reads, and also the low complex reads, like multiple A, multiple C, this kind of reads. So we get the reads as mappable reads. And then we just fill the copy number lower than 3 and the length. We try to fix this length to match the microRNA, like from 15 to 26. And this read will use for mapping purpose. So we map to the miR base and other like messenger RNA database, rna-seq, RNA family database, and the RAP base, and we try to identify what are these reads are. And then certainly we first find out what are no microRNA reads, and then what are no microRNA reads from other species like group 3, and then we also try to map this unidentified reads to the genome, specific genome for this sample, and try to identify which mapped region can form helping. So with this, we can predict what are the novel microRNAs which is not reported in any other database, like in miR base. So after this, we get know what we have in the plant, what the microRNAs in plant we can identify. And the next step that we try to identify the target of these microRNAs. So certainly we can first use the bioinformatics way, even without any experimental data. So we can do the prediction. So we can use that, as we know that the microRNA will be binding complementary, near perfect but complementary to the messenger RNA. So with this information, we can do a good bioinformatics prediction. And certainly we need to verify the prediction, or maybe even without any prediction, we can directly use the qPCR, qRT-PCR, and the Western blot, and the luciferase reporter assay to identify the specific target. But this method, these three methods are normally no throughput. That means that one-to-one identification, one microRNA to one target, or maybe a few. And we can go further. So bioinformatics, we know there's a few tools to do the prediction. And we need to keep in mind that the prediction normally still requires experimental confirmation, and also we know there's many false positive and false negative. And even we change different database, like rna-seq, if we use rna-seq or use other messenger RNA database, or maybe we use different microRNA database, we may get a different result from the prediction. And also different protocol, different program will give like a different prediction. So one thing I remember is that if you use different prediction method, normally they just share 40% for the same prediction, and the other 60% they will give the different result and different prediction result. So definitely we needed to use experimental method, as I mentioned, that we can use qRT-PCR and Western blot and luciferase reporter assays to verify the miRNAs target. But these are the low throughput method, and even for the RIS method, they are still gene specific. And the degradation sequencing is adopted from the RIS method, but they are high, but the degradation sequencing is a high throughput next generation sequencing method. So in plant, we know that the microRNA will cause cleavage of their target at the position between nucleotide 10 and 11 of the microRNAs. So, and we also know that the degradation sequencing will provide comprehensive means of analysis pattern of the RNA degradation. So we are not working on one microRNA or working on the one microRNA to multiple messenger RNA anymore. We are working on the whole degradome. We are working on the whole regulation of the microRNA on messenger RNA side. So next generation, we sequencing the 5' end of the RNA degraded products. So as I mentioned, that after the cleavage, the risks will leave 5-phosphate of the 3' end fragment of the messenger RNA. So use the degradation sequencing, we will identify what is exactly the sequence or the 5' end of that degraded RNA, messenger RNA, and then obtain this information, the 5' end RNA sequences about 35 nucleotide. We can map this back to the microRNA to see if they can match to any microRNAs which is supposed to be near perfect, complementarily mapped to the messenger RNA at this part. I will show a little bit more further in the following slides. So we did have our customer publish the I think about 10 or 15 papers right now from 2012, and I just listed a few papers here, and we will give some case studies, which is chosen from these papers. And first, we need a total RNA. So for our degradation sequencing, we only need a total RNA for this project, for the degradation sequencing. So customer need to purify the total RNA from their interested sample, and make sure that the quantity of the messenger RNAs, total RNA, is really good. And so after getting the total RNA, first we will, as I show in this flow chart, we will extract or maybe enrich the messenger RNA, which is supposed to contain the poly A tau, use the oligo DT magnetic beads. So as I show here. So the degraded messenger RNA do contain the poly-A tail and the 5-phosphate, right? So then after enriched by the oligo-DT beads, this will be ligated with the fiber adapter. So fiber adapter will ligate it to the phosphate only, phosphate 5' and only. And after that, we will have 5 adapter ligated degraded messenger RNA. And then after that, we will use 3' random primer to the reverse transcript. That means this random primer will reverse transcribe this messenger RNA. And after we obtain the first strand cDNA, we will use specific primers, PCR primers, to amplify the library. So after that, we do the size selection, and then we go by the gel purification, and then we do quantification, then we put on the sequencing. So normally we just need 10 microgram total RNA to do this using this modified method. And then we do use beads, magnetic beads, to do the selection, either poly-A or after the first strand, after the ligation. So the ligation is on the poly-A with the poly-A messenger RNA with the phosphate. And we do only 12, at the most 15 cycles of PCR, try to minimize the bias of the PCR. And we do only 1 purification. And the sequencing length just need 36 nucleotide. And normally after the purification, we don't have adapter contamination. So the whole experimental time will be 1 day, and or maybe 1.5 days, and it is compatible with the Illumina protocol or sequencing protocol. And so we get the sample prepared, and we, as normally Illumina protocol do, so we get a cluster generated, and we on the flow cell, and then we do the sequence and get the of the base call and the QC, and we get the row rates, and then we will do the data analysis after this step by LC science. Yeah. So the degradation sequencing, the major work we have done is that we are on the next generation sequencer. So currently we use GA2 for this degradation sequencing, and we sequence up to 35 bases, and we can get about 20 to 30 million reads per name. And the data output normally is 0.7 to 1 gigabases. And people sometimes want to do the barcoding, I mean the multiplexing. So for this degradation sequencing, we recommend normally it's just 1 or 2 samples per name, and the maximum number we could recommend is 3 samples. Why we recommend only 3 samples? Because we know that the degrading on the messenger RNA probably just a very snapshot of the whole transcriptome. And according to what we discovered, that normally just 20 or maybe 30, not more than 100 of reads can identify, which is mapped to the microRNA we can identify as a target reads, row reads. So if we put too many samples inside the sequencing name, probably this low abundant degraded messenger RNA fragment will not be discovered, or maybe not to be sequenced. So we need to have a very high coverage, or maybe high depths of the sequencing. So we need to have about 10 million reads to cover the degradome to identify the messenger RNA target. And we know that the sequencing name, the reads from the sequencing name will be fixed. They will not change. And you get more sample multiplexity, and you get less reads per sample. And for the degradome sequencing data analysis, we use we tried a few bioinformatics data analysis programs, and we choose after the comparison, we choose the Cleveland as our data analysis major program. And we also write some small Perl and Java scripts to make the result explained and also put it to the report for our customer. Try to make our customer easily to understand the result, data analysis result. So the data analysis flowchart will go like this. So first we use the degradome sequencing to identify the sequencing tag, which is we identify the 5' end of degraded messenger RNA, and then we map this sequencing tags, the sequencing reads, to the messenger RNA database, identify what are these fragments, sequence reads come from, and try to identify what are these messenger RNA. And then either come from the microRNA sequencing or from the miR-based database or other microRNA database. We combine the messenger RNA, degradome density and degradome, cleaved messenger reads with the microRNA reads, or maybe microRNA information. Then we can do the target prediction, and also from the data analysis, we know the abundance of each degradome, each degraded messenger RNA. So we will have a high fidelity messenger microRNA and the messenger RNA target pair, and we know the abundance and we know the cleavage site. So after degradome sequencing, our data analysis report will including Illumina-based calling and Illumina's row reads, and then we will map the degradome reads, the sequencing reads, to the messenger RNA database or maybe transcriptome, and then we will summarize the degradome density file, summarize the mapped degradome data, and to get a degradome density file. That means we identify the sequencing reads abundance, and also what are these reads come from, which messenger RNA it come from, and also go with the messenger RNA annotations. And then we try to generate the small RNA, microRNA, and the messenger RNA prediction. That's we identify the messenger RNA and the messenger RNA pair, and then we will compile the degradome density file to the target prediction, and then we also output the significant hit, and finally we will generate target plots, called T-plots, for the targets. So this table will first show what are the microRNAs we identified based on the messenger RNA degradome sequencing, and also come from our microRNA sequencing data or maybe database. And then we give the alignment score, and we will identify the alignment range, which is the location of the messenger RNA, and we know based on that the no information that the cleavage site is normally at 10 and 11, between 10 and 11 nucleotide of the microRNA. We can predict the cleavage site, and we give the category. I will give more detail about the category and the p-value, and we give the exactly how many row reads get from the sequencing, and also the reads of the normalization. If we want to compile, multiple samples, and then we give the annotation of the messenger RNA. And so we normally select the p-value less than 0.1, the pair with the p-value less than 0.1 in this table. And this is what I said is a t-plot. So the title will including what identify the microRNAs, and what the messenger RNA corresponding to the targeted messenger RNA and their cleavage site, and we also give the alignment score and give the category and p-value. So here, this read one stands for the reads we identified from the sequencing data, which match to the microRNA. So this means that we identify the reads is related to this cleavage site, related to this microRNA and the messenger RNA pair. And the black bars is also indicated the cleavage site of the messenger RNA, but it's supposed to not have the microRNA matched to this. So, for example, this read one, we get the row reads is about 18, and the others we still identify other cleavage on the messenger RNA side, but suppose we are not getting any microRNA pair to this. We didn't identify any microRNA, no microRNA for this cleavage site. Maybe they are coming from unknown microRNA, or maybe come from other cleavage happened to this messenger RNA. And the category 0 in simple explanation is that the reads we identified is the maximum reads on this messenger RNA. So and category 1 is category 0 is most apparently to be messenger RNA and the microRNA pair identified. Category 1 is that the reads abundance we found is equally to the maximum number for this messenger RNA. I mean, from here, this plot, you see there is two major cleavage sites identified from the degradome sequence, but only one got the microRNA pair, the pattern of the microRNA. And the category 2 is that what we identified is less than the maximum number. The abundance is less the maximum number. And the category 3 is that the category 2 is less than maximum, but more than medium reads we identify. And from degradome sequencing, category 3 is that they are less than the median number of the degradome sequencing reads on this messenger RNA. And the category 4 is only one row reads identified, so that's most unlikely to be the degradome and the messenger and the microRNA pair. But maybe it's due to the no total reads the no coverage. And for some studies, for example, I try to give some case studies. So for example, this is example 1. First, we have a customer identified the microRNA for this species, which is soybean, and we identify 97 known microRNAs and also 31 novel microRNAs. That means this 31 is unknown and is not recorded in the other database or other papers, and also we also identify some microRNAs which is coming from the same hairpin, but they are not reported. Maybe another strain of the hairpin structure. And after have a customer identify the novel and the known microRNAs, and we go further to do the Degradome sequencing. And then after data analysis, we identify 86 genes were identified as target of the known microRNAs, and 5 genes were found to be target of the novel microRNA obtained from the microRNA sequencing. So as we mentioned before, on the T plot, we are clearly see the cleavage site on the messenger RNA, like this one, and also we know what exactly the microRNA sequence played the regulation here, and we do have the experimental data, the Degradome sequencing to verify that we do find the degraded messenger RNA from this study. And this is category 0, and this is category 1. And so we also identify another one which have the row reads equal to the maximum number of the row reads we found for this messenger RNA. And this category 2, and this is category 3. So totally we identified, like I said, 86 genes regulated by the known microRNA and 5 genes regulated by the novel microRNAs we identified from the microRNA sequencing for these studies. And we also have the customer to identify the microRNA from the Oilseed, and we identify 41 conserved microRNAs, which is not known to the Oilseed and when the paper published, and but they are conserved microRNA, which is also found from other species, other plants. And also we have identified 62 non-conserved microRNAs, novel microRNAs for this specific species, and from the sequencing, microRNA sequencing, and later we have our customer to identify that 33 non-redundant target for the 25 conserved microRNA and the 19 targets, messenger RNA targets for the 17 specific microRNA, the non-conserved microRNAs for this species. So we are doing sequencing science 2008, and we are the major microRNA microRNA service company in US, and we also have an office in US and China, and also we have a sales representative in Japan, Korea, and India. And we provide our microRNA service, microRNA service science 2005, and we processed more than 15,000 samples, and the microarray platform we are currently using is our primary technical advantage, microfluidic, micro-paraflow, microfluidic technology. This is our own technologies, and we helped our customer about from the worldwide, and from 1,400 institutes and from 40 countries, and we get helped our customer publish more than 600 papers, and we, for the plant, we process almost like 80 kind of plant species, and we are good at handling plant samples. And yeah, this is our office and representative locations. Okay, thank you for attending our webinar, and if you got more questions, please go ahead to ask.